بصراحة المذاكرة شيء هذا لما اسوي
Okay, everybody take your seats. You know, we're already 5 minutes over the time, 5 or 6 minutes. So, remember that the DNA function, 3 things were important for DNA function. One was DNA replication, transcription, and translation. So, we've already talked about transcription, how we convert the DNA code into RNA. So today, we'll look at how that mRNA message is converted to protein, okay? So remember, protein is one of the molecules of life, and the cell makes, thousands of different types of proteins. So it's very active in translation. So remember, we get the mRNA message from the nucleus, okay? So transcription is happening in the nucleus, while translation is happening in the cytoplasm, in the rough endoplasmic reticulum, and in free ribosomes, okay? Within the cytoplasm, okay? So before we move on, remember I talked about a few types of RNAs that are involved in translation, okay? So messenger RNA, where's that coming from? As I said, from transcription, from converting the DNA sequence to RNA in the nucleus, okay? So messenger RNA. So messenger RNA, remember it's a single strand, okay? Does it have a ribose sugar or deoxyribose? Ribose, okay? And remember, the base pairs is IC. Instead of T, we have UC, okay? So that is messenger RNA. Then we have this transfer RNA. Where's transfer RNA made? Yeah.
Cytoplasm.
All RNA is made in the nucleus, okay?
Okay.
All RNA is made in the nucleus, okay? So you can't make it in the cytoplasm. So the transfer RNA is also made in the, all of the RNAs are made. So transfer RNA has a particular function in capturing amino acids, specific amino acids, and bringing it to the ribosome so it can add it to the growing amino acids to produce a protein. Remember that ribosomes are important for protein synthesis?
Yeah.
So these tRNAs are important in that function because they bring the amino acids to the ribosomes. We'll talk later about how that works, okay? Then you got the ribosomal RNA, which is also made in the nucleus, okay? So the ribosomal RNA, together with the ribosomal protein, remember we learned in chapter 3, make the subunits of the ribosomes in the nucleolus. You remember that?
Yes. Yeah.
In the nucleolus. So within the nucleus, the region known as the nucleolus is important for assembling the subunits, okay? Ribosomes are made up of 2 subunits, a small subunit and a large subunit.
Yeah.
So if you have a look here, there's the ribosome subunit, there's a small subunit and a large subunit. They're composed of ribosomal proteins, okay, and ribosomal RNA, okay? So RNA and proteins, ribosomal proteins. So as I mentioned, mRNA is actually a messenger between the DNA and the ribosomes, because the ribosomes can't read the DNA sequence, okay? they're outside the cytoplasm, okay? So there has to be a message sent to them. So the messenger RNA acts as that messenger between the DNA and the ribosomes. Then you have the tRNA, as I said, this is the small RNAs, they look like this. They structure. You remember the codons, the codon table?
Yes.
Yeah.
So if you look at the codon table, the genetic code, you see we have 64 different codons coding for the 20 amino acids. When we look at the structure of the tRNA,
Anticodon.
it has the anticodon, okay? Which is the complementary code for the codon. So if you have a look at here, if the codon sequence is ICC, what would be the anticodon? So it's the complementary. What's complementary to I?
U.
UGG. So it'll be UGG, okay? So the tRNA contains the anticodon sequence, and this is how it knows which amino acid to bring and to place in order. So when the ribosomes read the mRNA, okay, the ribosomes, they read it in order. So they will read this, they will read the next triplicate codon, then they'll read the next one, okay? They won't go read here, then come back here. It reads it in one direction. So it starts.
Had some methionine, I think.
Okay, so the ribosomes will read in one direction, okay?
Yeah.
It doesn't read it back, it doesn't skip, so it reads it one by one. And the tRNAs will come and bind to each triplicate, carrying the correct amino acid corresponding to that sequence, okay? And that sequence is based on this genetic code, okay? So remember, sometimes we can have up to 6 different codons coding for the same amino acid, okay? So the tRNA will have a particular amino acid that it captures from the cytoplasm, all right? So, and that amino acid is specific to that genetic code, okay? So, based on the codon sequence, it will have that particular amino acid, all right? If it has the wrong one, you'll get the wrong protein. So, it has to have the correct amino acid based on the genetic code, okay? So, once it captures this amino acid, it brings it to the ribosomes, and the ribosomes will join the amino acids as it reads. So as it reads the sequence, every time a tRNA comes and binds, it'll combine the amino acids that the tRNA is carrying, okay? So you see, have a look at here. The tRNA brings the amino acid with it. It binds to the complementary, okay? So you have a look at here. A is complementary to U is complementary to A, G, and C. So once it binds to the complementary base pairs, okay, the codon, then that amino acid is added by the ribosomes to the growing chain. You see the chain is growing as it's moving along. So as it's moving along the messenger RNA, every time it adds the amino acid based on the codon sequence, okay? And they'll just keep reading that. And the tRNAs will keep bringing in the amino acids. So that's the function of the tRNAs. They'll bring the amino acids to the correct spot, okay? To the correct sequence. And the tRNAs, once they bring the amino acids, and the ribosome has added it to the chain, it releases that tRNA, and the tRNA can go and get another amino acid, okay, from the cytoplasm and bring it. That's why it's called the transfer RNA. It brings the amino acids to the ribosomes, okay? Then you have the ribosomal RNA. This forms the subunits, okay? They combine with ribosomal proteins, they form the small and large subunits, which are important for translation, okay? And these are made in the nucleolus, as we know. So when we look at the ribosomes, they have sites for binding to the mRNA, okay? So they have regions in the ribosomes where they bind to messenger RNA, okay? And also they have regions where they bind to the incoming tRNAs, okay? So the ribosomes function in having the ability to bind to messenger RNA, okay? They have sites, and also binding to the tRNAs that are bringing the amino acids, okay? And that's important because it needs to bring all these things together to read the code and add the amino acids one by one. So all of these, the ribosomes have the capacity, the sites, to bind to messenger RNA first, okay? Once it does that, it also has the ability to bind to the tRNAs carrying the amino acids, okay? So it has binding sites for messenger RNA and tRNA, okay? It also has enzymatic activity, the ribosomes, because remember, how are proteins formed? Amino acids forming what sort of bonds? Peptide bonds, remember? Peptide bonds. So it has enzymes that catalyze the formation of the peptide bonds. So it does that by holding the messenger RNA and tRNA together. Okay, so in this case, here, how is it doing that? By formation of base pairing between the codon and anticodon. Okay, so the opposite complementary base pairs which are found on the tRNA. Okay, so that's what ribosomal RNA is for. Okay. Now, if you have a look at the actual process of translation, there is 3 steps involved in it, okay? If you have a look at the 3 steps, the first one is initiation, okay? Initiation means when you start something, okay? So we have initiation, then we have elongation. So lengthening something, elongating it, okay? Making it longer. How does it do that? By adding the different amino acids and producing the polypeptide, okay? So many amino acids joins together. So that is elongating the polypeptide, all right? That's called elongation. So then we have termination, where it stops, where it's the end of the protein, okay? So if you have a look at the initiation first, what happens is that the small subunit, okay, of the ribosome first attaches to the mRNA together with what we call the first tRNA. Remember the first AUG, the start of the protein? Yes. So which is methionine as well, okay? So the initiator, what we call the initiator tRNA. Initiator, it means it's starting translation, okay? Without the initiator, so the initiator tRNA, if you look at the next page, which carries the methionine, this is where it binds to the small subunit and the messenger RNA together. So what happens is the small subunit, the initiator tRNA carrying methionine, okay, binds to the messenger RNA, and then it will scan until it finds the start codon, okay? What's the start codon? AUG. And then what happens is that the large subunit then binds to it as well, okay? So then we have what we call the initiation complex, you know, because it has multiple things involved in it. We got messenger RNA, we got small subunit, we got tRNA, and we got the large subunit, all forming what we call the, is it written here? Initiation complex, okay? So these form the initiation complex. This is what triggers the start of translation, okay? This is what starts translation, when the small subunit, the tRNA and messenger RNA and large subunit bind together to form what we call the initiation complex, okay? This is what starts translation, okay? So that's the first step. This is called initiation, okay? Then the ribosome will scan, remember, in one direction continuously. This second step is elongation. So what happens is that tRNAs, remember, tRNA containing the anticodon will bring the specific amino acids corresponding to each codon, okay, each triplicate, and bind to the messenger RNA. As it's binding to it, the ribosome attaches the amino acids corresponding to that codon. So this happens, and the ribosome goes along the messenger RNA, okay? It keeps on doing it. It keeps on adding one after the other based on the code, okay? So it will do that until it reaches the end, okay? So you see, one after the other, different amino acids are joined together for this particular messenger RNA sequence, okay? Then finally, we have what is, yes?
So the large subunits bind the amino acids together, right?
Yeah, so it helps to form the peptide bonds, okay? The peptide bonds. It has enzyme activity to form the peptide bonds between amino acids. And it will do that based on the sequence, messenger RNA, one after the other, until it reaches the end, okay? So that is initiation. Elongation, okay? Then when you reach the end, we have the stop codon. Remember the stop codons?
Yes.
How many stop codons are there? Three stop codons. You see this table? We have 1, 2, 3 stop codons. If the ribosome hits any one of these, it will signal termination, okay? So in this particular sequence, we have a stop codon here, UGA, which is the stop. When the ribosome hits that, this initiation complex translation is terminated, and the initiation complex that was here, this complex, then disassembles, okay? The ribosomes, the messenger RNA subunits disassemble, okay?
What's the function for the small subunits?
Well, it brings these, they work together, okay?
Okay.
So the initial, where it binds to the mRNA and brings the tRNA, and then once it hits the AUG, this large subunit also binds. This together, all 3, 4, so 1, 2, 3, 4 form the initiation complex.
Then when they separate together also.
Yeah, they all separate out. When it hits the stop codon, it's saying that it is the end of translation, okay? Termination. So by that, so what happens is that the growing polypeptide that is made by the ribosome, so based on the mRNA sequence, is released, okay? And this then can be further modified. You know, remember we got modifications in packaging, modifications happening in the endomembrane system. Remember the endoplasmic reticulum, then to the Golgi. Proteins can be modified and refined and distributed, okay? If it enters the cytoplasm, it's needed for immediate use. Remember, free-floating ribosomes make proteins for the cytoplasmic use. So they become part of organelles, cell membrane, enzymes, and what the cell needs. Okay,
So
hormones and so on.
proteins are made in the cytoplasm, right?
Yeah, so they're made in the cytoplasm where they can either be attached to, the endoplasmic reticulum, the ribosomes, or free-floating ribosomes. Okay.
I mean protein.
Yes.
Protein are made in the cytoplasm.
Yes. Remember, there's two types of ribosomes.
Yes.
What?
And the
One rough endoplasmic
rough,
reticulum,
and
where
three
they
cells.
attach to the rough endoplasmic reticulum, and the other one they're floating in the cytoplasm. Okay. If we have a look at the cell, this was the question in the exam. Remember?
yes, I remember.
The tubular. So once these peptide chains or polypeptides are made, they can be further refined, modified, and shipped to locations, or they can become part of the cytoplasm. And proteins, remember, we find in the plasma membrane lots of proteins for different functions. You remember chapter 3? We have proteins for channels, we have transport proteins, receptor recognition proteins, enzymes. Or we could have proteins in the cytoplasm, like the cytoskeleton is made up of actin, tubulin, okay? These are proteins. Or you can have other enzymes or hormones sometimes, like insulin is a protein, okay? So there is different functions with these peptides, and they can be directed to different places in the cell. So any questions on translation? Yes.
I didn't get the function of tRNA.
So tRNA, transfer RNA, okay? If you have a look at it here, its function is it carries the amino acids. What is protein made up of? Amino acids joined together. How are the amino acids brought to the ribosome so they can join them together? The tRNA does that, okay? The tRNA transfers the amino acids to the ribosomes. That's why it's called tRNA, transfer RNA. So the tRNA brings the amino acids to the ribosomes based on what? Because the tRNA has the anticodon sequence, okay? Remember the genetic code? Here? The tRNA contains the complementary sequence of that. So how does it know which? So it carries, based on this sequence, okay? It carries the amino acid, okay? For example, if we have a tRNA with the sequence, the anticodon sequence IUG, okay? Which amino acid will it be carrying?
UAC.
So
No.
UAC, which one is UAC? Huh? So I'll be carrying tyrosine, okay? You got that? So I'll be carrying tyrosine if it's got the anticodon sequence for that.
So the initiation should start with.
Sorry. What did I say?
Doctor. So the initiation should start with AUG and UAC.
So if it's carrying AUG, the anticodon, should be UAC. What would be the anticodon for this initiation? For IUG, what is the anticodon sequence?
UAC.
UAC, okay? So if the tRNA contains UAC, which amino acid is it bringing?
AUG.
AUG is not the amino acid.
Methionine.
Methionine. Did you understand what's happening?
Yeah.
Anybody else confused? Yes.
Why?
How is this going to recognize the message? Yeah. So it has to be complementary to it, correct? Base pairing. So if it's A, what's the complementary anticodon?
T. U.
U. So U, it has to be I, G, C. Complementary base pairing, remember? Yes.
From where you come?
U is RNA, okay? In RNA, remember we have DNA has 4 nucleotides. ITGC, RNA, IUGC. So the complementary base pairing is very important here, okay? And that's how the tRNAs work. So the tRNA has the anticodon sequence for IUG. IUG codes for what? Start methionine. So if a tRNA has the code UAC, what amino acid is it carrying? Yes. What amino acid is it carrying?
Which one?
Methionine. You see here? It's carrying methionine. So what would be the anticodon sequence of the ICU? So it should be UGI. Okay, let's have a look at here. What amino acid should it be carrying if it's UGI? But because it's if the anticodon is UGI, the codon sequence is ICU. ICU. This genetic table works on the codon sequence, not anticodon. Don't confuse it, okay? If I give you the anticodon sequence, you have to convert it to the codon before you can read it off this, okay?
I don't understand.
If I give you this sequence, I said a tRNA with the anticodon sequence UAC, which amino acid is it carrying?
So you have to
Convert it to the codon sequence. How do you do that? The complementary base pair, okay? So it should be IUG. You got that? Then you would tell me, based on the table here, that it is methionine. If I say I gave you a sequence ICG, the anticodon, which amino acid is it?
ACG.
ICG.
UGC.
UG.
C.
UGC. So what is it?
UGC to
Cysteine. Okay? You understand how it works? You have to convert the sequence to the anticodon. So if I gave you the tRNA sequence, the anticodon sequence, IUG, you convert that. What's IUG converted to codon? The complementary base pair. Do you know? IUG? IUG, so UAC, huh? So what amino acid is UAC? In the table, if you read the table, which amino acid will be UAC? UAC. So remember, first U, then we look for the, the first one U, then the second one I, then C. So UAC. Tyrosine, okay? Do you remember how to read the table? So you look for the first letter, then you look the second, then the third. Okay? That's how you find the very easily the codon that you're looking for. Yeah.
So, Doctor,
You don't need to remember this table, we'll include it.
Here will be third. Here will be third. Right.
So ICU, if you have a look at it, where's ICU? So we got I, C, then we got U. So it's threonine, yeah. If I gave you the anticodon sequence of that, okay? So if I gave you UGA, you first have to convert it
to the
codon, then tell me what amino acid it is, okay? So you got codon and anticodon. If you have a look at here, see, if you have a look at here, if the codon is ICC, the anticodon is UGG. So U complementary to I, G complementary to C, G complementary to C.
But what's being read is the codon.
Yes.
So we take the amino acid of the codon.
Yeah. But if I give you a question in the exam and say, The anti-codon sequence is this. Which amino acid is it carrying?
So you should convert it to
Convert it to the codon
codon,
if you
then-
start. If you read that from the table,
So
from the anti-codon,
mistake.
you'll get the wrong sequence. Okay?
Wait, can you
Yes.
repeat the question?
If I gave you the anti-codon sequence, okay, and I told you to give me what amino acid does that code for?
Also converted to code.
First convert it to the codon, then read the table, then tell me what amino acids, okay? Yes.
Would you do the opposite? Like give us the amino acid and then you're like, what anticodon is this?
We can do that, yeah, but usually
anticodon don't give us.
yeah, you can. We can give you the amino acid and give you several different sequences, and that way it'll be a bit more difficult because there's different combinations, but you can figure it out based on the sequence. Okay. If I give you the amino acid and I give you a list of 4 different options with different mRNA sequences, okay? You just need to convert all of them to the amino acid, then you'll know which one it is. If you get a question like that, okay? Convert it to amino acid, then you can say, oh yeah, this is the correct one. Because you remember, one amino acid can be coded by many codons. You see serine here?
6.
All of these code for serine, okay?
Yeah.
All right, let's have a look at this activity here. All right, so we have another one of these analogies. Remember comparing, in this case,
Cities
to other things, okay? In this case, we're comparing the gene expression to cooking, okay? So, using the cooking analogy for gene expression, which one carries out the following functions, okay? The one that brings the recipe. So if we talked about gene expression, remember it involves transcription and translation, okay? So the one that brings the recipe is what? Think about it. The one that brings the recipe. You know what a recipe is?
DNA?
mRNA.
mRNA, okay?
Yeah.
What's the recipe?
Recipe, the instructions on how to make the, you know? So, where are the instructions? Yeah, so mRNA, or in this case, I mean, it's gonna be the mRNA because it's bringing the instructions to the ribosomes, alright? So, it's mRNA. The one who finds and delivers the ingredients, the one that brings the ingredients, the tRNA, obviously, because it's bringing the amino acids to the ribosomes. So the ingredients of protein is what? An amino acid. So tRNA here. The cook who creates the final product, the ribosome. Okay. So the ribosomes are the one that joins the amino acids together. The ribosome is the cook. Okay. So he's putting, or the ribosome is putting all the
Together, bring them together.
the ingredients together to make the protein, all right?
So ribosomes or ribosomal RNA?
Ribosomes.
Not the ribosomal RNA.
Well, they're all together, working together.
The
It's
same
a-
thing?
yeah. Ribosome are formed by ribosomal RNA and protein.
Yeah.
The subunits, okay? It's working together.
So what's the
ribosome?
for the food?
This one is ribosome.
Ribosomes are formed with ribosomal RNA and protein.
Yeah. So ribosomes.
And I mean the ribosomes.
Yes, ribosomal RNA and ribosomal protein. They make the subunits of
Okay,
the
and the
ribosome.
nucleus, yes.
In the nucleolus.
Nucleus.
All right. The cell spends most of its life in what phase?
Interphase.
Interphase. Okay. Remember the cell cycle. Most of the cycle is an interphase, like G1, S, G2. Okay. Sometimes they spend all their life maybe in that G0. Okay. So they never go into the mitotic phase. So they spend most of their time in interphase. Only a short period is in cell division, which is the rest of these phases, you know, the M phase or the prophase, metaphase, which is wrong, okay? Alright, name the following structures. Which structure do you think is A?
Homologous.
Homologous chromosomes, huh? Homologous A? Homologous chromosomes?
Yeah.
Yeah. Yep. So B, these two. What are they? Sister chromatids, okay? Sister chromatids. So B is sister chromatids, okay? Remember, when the chromosome is duplicated,
Yes.
the duplicated chromosomes have the two sister chromatids.
But doctor, when they separate, we don't call them sister.
No,
sister
individual
chromosomes.
chromosomes.
It will be chromosomes.
Yeah, but in
So
this
the-
case, it's sister chromatids in this form.
So we see chromatid only if they are together,
Yeah.
replicated.
Sister chromatids. Alright, C, what is that structure? C is the centromere. Remember, the centromere joins the sister chromatids together.
Yeah.
So C is the centromere. D, what do we call that? Huh? What's that? Those two?
Sister chromatid.
Crossing over happens between what? Yeah, in prophase between what?
Between two
Non-sister chromatids, remember? So these are non-sister chromatids, okay? Because they're from different chromosomes, okay? So they're non-sister. These two are sister. These two are non-sister. Okay? Yeah. What about E?
sister chromatid.
Well, to me, whoever made this activity, it looks like non-homologous chromosomes.
Non-homologous.
They're not homologous, okay? That's what I think about it. Yeah, because this is not homologous to that chromosome. Remember homologous chromosomes from the mother? For each, we have 23 homologous pairs. So this chromosome is not homologous to that.
An exam you can give us like.
Nah, you're not gonna get this in the exam.
There is no figures in the exam.
Huh?
There is no figures in the
No,
exam.
there's no figures. Just like this questions. Exam is all multiple choice.
So what's
E is non-homologous chromosome. Non-homologous.
Count. No.
What's the time?
7 times.
Question, the third one, what is the answer?
Which one?
Which one? Here? Ribosomes.
Why is not RR? I think the same.
What makes the ribosomes? The ribosomal protein and the ribosomal RNA together. Okay? Ribosomes are made from ribosomal RNA and ribosomal proteins. Okay, let's see if we have a Kahoot here. We can do it in the next 5 or 6 minutes. Did we do Kahoot on